All right, let's look at how genes are moved between the species to make recombinant DNA and transgenic organisms. So one of the earlier research techniques in this area was when we were able to isolate the green fluorescent protein, GFP, and it's found in jellyfish. It's what makes them fluoresce like this. You're able to take that particular gene that's responsible for making that fluorescent protein, because remember our genes code for certain proteins, and put them into another organism such as these mice. And you can see that where the genes are expressed, they are fluorescing, whereas the mice that do not have that gene expressed or inserted are not fluorescing. This was just to find out if they could do it right here. But over here, you can also use it to find out which genes are turned on. So if you put that fluorescent protein in certain pieces of DNA, you can find out what it does. Here's an embryo right here, and you can see where they put the this recombinant DNA, where they put it in the genome will tell you if the gene that they inserted it into is also active. So if certain pieces are fluorescing, then that tells you that part of the gene is active. So that's pretty cool. It's a way to tag certain genes to find out what they do. Okay, so you can also use recombinant DNA and let me show you what I was talking about earlier, transgenic organisms. It's hard going between slides here for you guys with different videos. Make sure you email me if you have any questions. So you can also make something called, well, genetically modified plants. So genetically modified plants don't always have a foreign gene inserted into them. Sometimes they are just bred. They're selectively bred, and that will that will modify their genes as well. However, there are plants, and this one right here is cotton, that have foreign genes inserted into their genome. So this is BT cotton, and it was created because of this bollworm here. So bollworms used to just devastate, as you can see over here, the cotton crops. I think this is in India, and it just used to devastate all their cotton crops. And so they inserted a gene into this cotton called BT cotton that would produce a toxin so that when the bollworm would eat it, the bollworm would die, and hopefully save this cotton crop. So I think this was Monsanto product, and they went and made a whole bunch of this. They sold it over to, I believe this is India, and it worked for a few years. And then, guess what? This bollworm evolved so that it was not hurt by this toxin, because any of the bollworms that survived this toxin that was created by the BT cotton then went and had babies and evolved to be immune to this toxin here. So it didn't work very long. It actually created this super bollworm that's now really hard to kill, and it made it even worse over there. So worked for a minute, and then in the end actually made everything even worse. Now that bollworm's even harder to kill. But that is an example of these foreign genes being inserted into a crop. So there are soybeans that are resistant to common herbicides, like Roundup Ready soybeans, for example. And then some plants are actually being engineered to produce human proteins, like hormones, clotting factors, and antibodies into their seeds so that they can extract medicine from them that way. So those are a few uses of genetically modified crops. Instead of inserting genes, sometimes you can just silence a gene or break it. So if you're able to break a gene, then you turn that gene off. For example, this protein is responsible for the apple turning brown. You know, when you cut an apple and it's exposed to the oxygen in the air, it will actually turn brown after a while. They have some apples now that gene has been silenced in, and they do not turn brown when you cut them. So kind of neat. Not sure it was necessary, but pretty cool for the Apple industry. Okay, let's look at some genetically modified animals. So I don't know if you guys have heard of glowfish. They're pretty cool. There's actually a whole bunch of different types of glowfish now. There's a barb shark and let's see, there's tetras. I can't remember what all there is, but they're pretty cool. You can find them at PetSmart. And they've actually got the fluorescent jellyfish gene that has been inserted into their eggs. And then you've probably heard of the bovine growth hormone. Some salmon farms are using that so that they can produce salmon that looks like this back one. It's huge. And bovine growth hormone, that means it came from bovine, or the cow industry. So the cow hormone, growth hormone, was put into the salmon to make huge yields. So there's also an industry for transgenic animals called gene farming. Gene farming with a PH. It's the use of transgenic farm animals to produce pharmaceuticals. It actually produces it in their milk so that the animals will produce the protein and you can extract it from their milk. There's plans to produce drugs to treat cystic fibrosis, cancer, blood diseases, etc. You're not seeing it very widely used right now. Okay, let's look at gene therapy. Gene therapy involves procedures to give patients healthy genes to make up for a faulty gene. So if there is a broken gene in a genome, then you can actually cut out a healthy gene from another organism and introduce it into the genome that contains the broken gene. There's two ways to do this. One way is ex vivo, meaning outside the body. And so you can actually take bone marrow stem cells and infect them with a virus that carries the gene for ADA, adenosine deaminase. So it's an enzyme that they're lacking. So you can actually put it in their bone marrow stem cells and then reintroduce it back into the body. In this case, a lot of children with severe immunodeficiency. Another way to do it is to do it inside the body, in vivo. So, for example, in cystic fibrosis, you can actually insert p53 tumor suppressor gene into cancer cells to make tumors responsive to chemotherapy. That's separate. That's for cancer. They're also working on something with cystic fibrosis. But they can actually just insert it directly into the affected area of the body. And this is showing an example right here where this is ex vivo. You take bone marrow and there's a defective gene that's cut out, and then you can put in a retrovirus which has the correct form of the gene in it, and it inserts into the genome so that you have now a normal gene, and you put it back into the bone marrow where your body's going to copy and make many copies of that correct gene. Okay, and we have a few more minutes, so I'm going to show you the different fields in science right now that are working on this biotechnology. And these fields are really expanding right now. This is kind of the new frontier here. So the old school way was just called genomics. For example, the Human Genome Project. This was a huge project worked on for 13 years to sequence 3 billion base pairs. And it wasn't just one company or one university or one group of scientists working on it. It was the whole world. So we used universities and private labs throughout the whole world. And in 13 years, they were able to find every single sequence for those 3 billion base pairs in human DNA. So that's a pretty huge project. And everybody thought, wow, now that we know the sequence of DNA code, then we're going to be able to solve all the world's problems, right? But that's not how it worked. We found out that it's actually a lot more complicated than we first realized. So humans have between 21,000 and 23,000 genes. Most code for proteins, and that's just genes. So not all of the DNA codes for certain genes. But most of those genes do code for proteins. 90% of the average protein coding gene in humans are introns. Those are the ones that are in the way. Much of the human genome was formerly described as junk DNA, but we're now finding out that there are a lot of uses for this junk DNA. It's not actually junk. That's that repetitive DNA that doesn't code for a physical trait that we see. But if we look at, so when we went ahead and found out the base pairs, that's called structural DNA. That's knowing the base sequence. But now that we found out it's a lot more complicated than just the sequences, there's a new field known as functional genomics. So what do those sequences do? And they found out that the express genes, the genes that actually express certain traits, the coding DNA that we've been talking about, that's only 2% of your base pairs. The rest are mostly repetitive DNA sequences and introns, and then this unique DNA. So those introns, just to remind you, here's exons. These are the actual coding DNA that code for certain traits. These introns were those pieces that were in the way. When we cut those out, those actually do have uses, which we can't get into right now, but those are important. And also remember, those exons can be put together in different ways to produce different genes, even though they're parts of the same sequence. And then you have this repetitive and unique DNA. This is what we can use to look at DNA fingerprints, to compare crime scenes and things like that we talked about earlier. Okay, so functional genomics aims to understand the role of the genome in cells or organisms. We can make a DNA microarray that can monitor the expression of thousands of genes simultaneously and tell us what genes are turned on, environmental conditions that turn genes on, and you can also look at what sorts of diseases you are predisposed to. For example, the company 23andMe, this is their model. This is what they do. They look at how likely a person is to have a certain disease sometime in their lifetime based on their DNA, and that's done with a DNA microarray. Check them out. You can actually look them up online. Okay, so this is what the DNA microarray looks like. You'll actually have the subject's DNA here, and then the subject's DNA is going to bind to certain parts that code for certain diseases, and it'll light up on the screen. Pretty amazing. It can get your genetic profile. It indicates any genetic illnesses and what type of therapy might even be appropriate as well. There's also a field called comparative genomics where you can compare the genomes of different organisms. And then there's something called proteomics, and this is where the frontier is right now. This is where the big research is going on right now. Proteomics. Because we're looking at the actual proteins that are coded for from the DNA and what the proteins do, how they're folded. This is super complicated. Proteins fold into these super complicated structures, and there's many parts to them, not just one involved. So this we were not able to look at before very well because we didn't have the computing power. Now we do have computers that can look at this, and that's where we use bioinformatics. So this is the new frontier, huge deal. My husband is actually a software engineer, and he has put this onto our computer. Let me show you. It's called Folding at Home. Let's see if I can get it to come out here. I guess I can't. It's called Folding at Home. So what it's able to do, I wish I could make my screen bigger. What it's able to do is use the background of your computer that you're not using the computing power for. It doesn't slow down your computer at all. Here's the website for it, Client Folding at Home. And it will use your computer to run in the background and help figure out what these proteins do for proteomics. Right now mine's starting a new one, so it's at 0%, but it shows you what project you're actually working on. All week it's been working on COVID drugs actually and how it targets different proteins. So right now it looks like it's doing something else, but this is a neat site to check out, Folding at Home. All right, that's all of my time. I miss you guys so much, and let me know if you have any questions. This is one of my favorite chapters. I wish we could have been in class together, but I am here for you if you need anything. Just let me know. Thanks, guys.